Hello friends, today we are here to discuss about a very important system of physiology in our body. Friends, you all know that cells in our body require various substances such as oxygen and nutrients. Also, the cells produce waste products that have to be removed. Can you now tell me which system is responsible for such transport of substances? Yes, it is the circulatory system. So students, today we are going to talk about the chapter Body Fluids and Circulation. We are going to divide this chapter into two parts, and today we are going to cover the first part, which is body fluids. Friends, you must have already studied about the various types of tissues that are present in our body. Can you name those tissues? They are of four types: connective tissue, epithelial tissue, muscle tissue and nervous tissue. Now do you think blood is a type of tissue? Yes, students. Despite being fluid in nature, blood is a connective tissue. You must be wondering, why is it called a tissue? There are two reasons. Firstly, it has the same origin as that of the other connective tissues like tendons, ligament and cartilage. And secondly, it connects the different systems in our body by transporting substances in and out of cells. So, blood is the softest connective tissue in our body. You must already be aware of the concept of pH and the three different ranges of pH. Neutral pH is 7, above 7 is alkaline and below 7 is acidic pH. Blood has a slightly alkaline pH of 7.4. Now, can you take a guess how much volume of blood is present in your body? There is 5.5 liters of blood which is present in a healthy adult. Let us now discuss about the composition of blood. Blood is composed of plasma and formed elements. As you can see in the pie chart, 55% of blood is plasma and 45% of blood is composed of formed elements. Now, there are different types of formed elements. What are these? These are the red blood cells, the white blood cells and platelets. So, if you draw blood from an individual and keep it in a test tube, you would observe it as a red fluid. But when you centrifuge it, you will see three layers. The topmost clear layer, which is pale yellow in color, is of plasma. Below it, the white interface layer is of white blood cells and the lowermost red layer is of red blood cells. So, let us talk about the composition of the first layer of plasma. It is composed of mostly water, which is around 92%, and a small proportion of proteins, which is 8%. If we follow the chart of detailed composition of plasma, we can see that it is composed of water, ions such as sodium, calcium, magnesium, bicarbonate and chloride, organic molecules such as amino acids, proteins, glucose, lipids and nitrogenous wastes, and gases such as oxygen and carbon dioxide. We have already discussed that out of all these components, water and proteins are the most abundant. Proteins of the blood are fibrinogen, globulin and albumin. Students, you would be amazed to know that the role of blood is not just to transport substances. You must have observed that when you get an injury which involves a blood loss, there is a formation of clot at the site of injury after some time. The protein that is responsible for this clot formation is fibrinogen, which is a part of blood plasma. The globulin protein present in plasma is responsible for mediating defense mechanisms, and albumin protein helps in the maintenance of osmotic balance. Friends, once again, if we take some blood in a test tube and allow it to stand for some time, we observe a clot at the bottom of the tube, as you can see in the diagram. The clear fluid that remains on the top of this clot is known as serum. So now the question arises, what is the composition of this serum? Students, if fibrinogen or clotting factors are responsible for clot formation, that means they will be present in the clot. So the composition of serum on its top is plasma minus the clotting factors or fibrinogen. Let us now quickly enlist all the functions of plasma. Plasma helps in clotting of the blood. It helps in retention of water in blood. Immunoglobulins present in plasma help in providing immunity. Plasma proteins help in maintaining the pH of blood, therefore act as buffers, and it also helps in transport of materials. So, till now we have discussed the composition and function of blood plasma. Now we shall move on to formed elements of blood that comprise 45% of the blood volume. The formed elements are composed of erythrocytes or RBCs, leukocytes or WBCs, and thrombocytes or platelets. Erythrocytes are also known as red blood corpuscles and are the most abundant cells in human body. Around 5 to 5.5 million RBCs are present per cubic millimeter blood in our body. In humans, the RBCs are devoid of a nucleus and are biconcave and circular in shape, while in camels they are nucleated and oval in shape. Students, cell has evolved from a more simple non-nucleated type to a complex nucleated type. So, do you think by having lost the nucleus we are more primitive? No, by losing the nucleus we have actually become more evolved, because now the RBCs can carry more hemoglobin, which we are going to discuss later. But for this we have paid a price, as the life of RBCs is shortened to only 120 days. Friends, can you now tell me what is the synthesis of RBCs known as, and where are they synthesized in our body? Synthesis of RBCs is known as erythropoiesis. In early embryonic life, erythropoiesis takes place in the yolk sac. In later embryonic life, this is taken over by liver, while in the adults it takes place in bone marrow of long bones such as femur and humerus, and in flat bones such as sternum, pelvis, ribs, skull and vertebrae. Students, there is a fixed number of RBCs present in our body, but in case of certain diseases, this number can go up or down. If the number of RBCs drops down, the person is said to be suffering from anemia. As you can see in the figure, the RBC layer in the test tube becomes less than normal. In case of polycythemia, the number of RBCs becomes much more than the normal condition, which can be seen as a bigger red layer in the test tube as shown in the figure. Now, how do we determine the number of RBCs in an individual? This is done by cell counting. For counting the number of RBCs, a special slide known as the Neubauer's chamber is used, on which blood are counted on two square shaped ruled areas. One of these two areas is zoomed in for better understanding in this figure. We put a cover slip on the center and then we put a small drop of blood on the slide, which seeps in between the slide and the cover slip. As you can see, there are 9 squares in the counting area. The central square is used for counting the RBCs. Within the central square, there are 25 smaller squares. RBCs are counted in the 4 small corners of the central square. RBC counting squares are marked as R in the figure. So, using this chamber, we find out whether the number of RBCs is normal, less or more. Now, students, can you tell me why does the blood appear red in color? It is mainly due to the presence of hemoglobin in the blood. RBCs are devoid of a nucleus and other cell organs, so that they can carry more hemoglobin. 12 to 16 grams of hemoglobin is present per 100 ml of blood. The function of hemoglobin is to carry respiratory gases in our body. Oxygen from lungs is carried by hemoglobin to the tissues where they release this oxygen. Now, hemoglobin has two components. One is the globin protein and the other is a red pigment known as heme. Heme is an iron porphyrin complex and one hemoglobin molecule has 4 hemes in it. Alteration in the structure of hemoglobin is associated with genetic diseases such as thalassemia and sickle cell anemia. In thalassemia, there is a defect in the globin gene of hemoglobin which leads to deformed RBCs. Patients often require blood transfusions in this case. However, in sickle cell anemia, the RBCs become sickle-shaped and have reduced capacity of carrying oxygen. Now students, can you tell me the age of a red blood cell? An RBC has to travel long distance all through the body, during which there is a constant wear and tear. The average lifespan of an RBC is 120 days, after which it undergoes degradation in spleen, which is also known as the graveyard of RBCs. After RBCs, let us now discuss about the WBCs or white blood corpuscles. WBCs are colorless because of lack of hemoglobin. They are much less in number as compared to RBCs. There are 6,000 to 8,000 WBCs present per cubic millimeter of blood. Not just the number, even their size is smaller than RBCs. They are round or irregular in shape, and unlike the RBCs, they contain a nucleus and other cell organelles. Now, depending on the presence of granules in their cytoplasm, WBCs are classified as granulocytes containing granules and agranulocytes which do not have granules in their cytoplasm. Granulocytes again are of three types: neutrophils, basophils and eosinophils. Whereas agranulocytes are of two types, monocytes and lymphocytes. We shall discuss each of the cell types one by one. Let us start with the three granulocytes. Neutrophils are the most abundant of all the WBCs, that is around 60 to 65% of total WBCs, and as you can see in the figure, they have a multi-lobed nucleus. They are weakly stained with acidic and basic dyes and are phagocytic or engulfing in nature. Eosinophils are 2 to 3% of WBCs. They have two-lobe nucleus and they get their name because they are stained red with acidic stain eosin. The number of eosinophils gets increased or decreased in case of allergies. The number of eosinophils get increased in case of allergies. Basophils form the smallest percentage of WBCs, which is around 0.5 to 1%. They have a 2 or 3 lobed nucleus, and basophils get their name as they are stained with basic dyes such as methylene blue. They cause release of serotonin, histamine and heparin during inflammation of a cell. Agranulocytes, if you see the image, have much less or no granules as compared to granulocytes. And as already mentioned, they are of two types, monocytes and lymphocytes. Lymphocytes again are of two types, B cells and T cells. They have a large nucleus with a very little cytoplasm. They are involved in antibody production, graft rejection at the time of organ and tissue transplantation, and they are non-phagocytic in nature. Monocytes are larger in size and have more cytoplasm inside them, and the shape of the nucleus is bean-shaped. Monocytes are phagocytic and they engulf cellular debris, foreign substances and microbes, etc. At the time of an injury or wound, monocytes move to the site of injury through the bloodstream. Once they enter the appropriate tissue, they differentiate into macrophages, which then perform the function of phagocytosis. Friends, after RBCs and WBCs, the third component of formed elements is platelets. There are approximately 1.5 to 3.5 lakh platelets present per millimeter cube of blood. Now, platelets are fragments of cytoplasm and they do not have a nucleus. They are formed from the large cells in bone marrow known as the megakaryocytes. You must have heard that in certain diseases like dengue, the number of platelets falls and the patients require blood transfusion. The primary function of these platelets is to release various factors that help in clotting of the blood. Friends, blood clotting is also known as coagulation of blood. In this process, there is a cascade of reactions that takes place which leads to the formation of a reddish brown clot which you all must have observed during an injury. Now, there are two pathways by which clot formation takes place. These are the extrinsic and the intrinsic pathways of clot formation. In the extrinsic pathway, there is damage outside the blood vessel, which causes release of tissue thromboplastin. In the intrinsic pathway, there is damage to the blood vessels that activate the cascade of clotting factors. Both the extrinsic and the intrinsic pathways help in the activation of factor 10. In the presence of calcium ions, active factor 10 converts prothrombin to thrombin. Thrombin is responsible for conversion of fibrinogen to fibrin. Now, fibrin along with a factor 13 forms a clot or mesh over the wound. Once a clot is formed, bleeding stops and the clot hardens. New cells form to repair the site of injury, and slowly the clot becomes smaller and it is ultimately dissolved by an enzyme known as plasmin. So, now you know that a whole lot of reactions takes place when you get even a small cut or injury. We shall now move on to discuss about the various blood groups that we all have. Can you now tell me what exactly is meant by blood groups? Friends, blood cells have carbohydrate molecules on the outside. A specific pattern or chain of specific polysaccharides determines the different blood groups. For example, every individual has a basic chain of H antigen on the surface of RBC, which has the sequence glucose, galactose, N-acetylglucosamine, galactose and fucose. As you can see in the figure, if the galactose present before fucose has N-acetylglucosamine attached to it, it becomes A type. If another galactose moiety is attached to it, it becomes B blood type. And if no group is attached to this H antigen, it is O blood type. So, there are two antigens A and B. If an individual has only A antigens on his or her RBCs, he or she is of A blood type. If only B antigen is present on RBCs, it is B type of blood group. If both A and B antigens are present on RBCs, it is AB blood type. And if no antigens are present on the RBCs, it is O blood type. Now, all these blood types have different antibodies present in their blood. For example, A blood type has antibodies against B blood type, which are known as anti-B antibodies. B blood type has anti-A antibodies, AB blood type has no antibodies, while O type has both anti-A and anti-B antibodies. A type can receive blood only from A or O type of individual. Similarly, B type can accept blood from another B type or O type. AB type has no antibodies, so it can accept blood from all types of individuals. Hence, AB type is also known as the universal acceptor. O type can only receive blood from another O type, however, it can give blood to all other blood types. Therefore, it is known as universal donor. Blood type must be matched before transfusion, as mismatched blood leads to clumping or agglutination. So, now can you tell me what exactly happens in clumping or agglutination of blood? Let us take an example of a person with A type of blood. If A type individual is given blood from a B type of individual, the anti-B antibodies of A will react with the B antigens, causing clumping and destruction of RBCs, also called as hemolysis. Therefore, A individual can accept blood from another A individual or O individual only. You must have also heard about positive and negative blood groups. Now, what determines positive or negative type? Friends, just like the H antigen which determines the different blood types, there is an Rh antigen which is responsible for positive or a negative type. Rh antigen is also present on the surface of RBCs. If Rh antigen is present, the person is said to be Rh positive, and if it is absent, the person is said to be Rh negative. Now, an Rh negative person exposed to Rh positive blood forms antibodies against Rh antigen. So, Rh typing must also be done before blood transfusion. Rh incompatibility problem is faced during pregnancy that may lead to erythroblastosis fetalis. What is Rh incompatibility? During pregnancy, if the mother is Rh negative and conceives a fetus that is Rh positive, the cells from Rh positive fetus can enter the mother's bloodstream, and the mother develops anti-Rh antibodies to fight these Rh positive cells. This is known as sensitization. If the sensitized Rh negative mother conceives or becomes pregnant with an Rh positive fetus for the second time, the anti-Rh antibodies of the mother attack the RBCs of the fetus. This is known as erythroblastosis fetalis, which can even cause death of the baby. To overcome this problem, anti-Rh antibodies are administered to the Rh negative mother immediately after the delivery of first child. Friends, we shall now move on to another body fluid called as lymph. Unlike blood, lymph is colorless. Students, blood flows through high pressure in the capillaries. Because of this high pressure, some amount of water along with solutes seeps out in between cells of tissues. This fluid is known as lymph, which is also referred as interstitial or tissue fluid that bathes all the cells. Lymph causes exchange of nutrients, gases and hormones between blood and cells. It also contains lymphocytes, about which we have already discussed. Another very important function of lymph is absorption of fat in lacteals of intestinal villi. Unlike all the other nutrients that are absorbed by blood in the small intestine, fat is absorbed only by the lymph. With this, we have come to the end of today's topic. We have talked about the major transporting agent blood and its components plasma and formed elements, various blood groups, Rh factor, coagulation of blood and another body fluid, lymph. In the next lecture, we shall discuss about the human circulatory system in detail.